Okay. So I'm going to kind of pick up where I left off before and talk about current soil testing because it gives a foundation. So I'm also getting text messages about the link, by the way. But current soil testing treats soil as a non-living, non-integrated system. We use harsh chemical extracts to try and figure out what's happening in your soil with the chemical system. So current soil testing focuses mostly on physical and chemical and not the biological aspects, which if you're trying to move into soil health, we really want to know what's happening in the biological part. The extracts that most labs used were formed or developed between 1945 and 1984. This picture right here is an old tractor. I don't know how many of you want to run that right now. Russell, my business partner, did for a very long time, had a very open cab, loud tractor. So we all want to move forward in what we're doing and do things the best way we possibly can. And so that's why we developed the Haney test. And I'm hoping you can see my screen. Yes? Okay.
Yeah, you're good, Liz.
Okay, good. So we're trying to mimic how the soil responds after rainfall in your field with plants growing, and not how it responds to these artificial methods and chemistry that we have created in the laboratory. When agriculture obviously has been going on since a very long time, but after the Industrial Revolution, we really started focusing on a very limited focus, reductionist way of thinking. We apply NPK, and this is how the plant grows. But biology is much, much more complex, and that's what we're trying to mimic in our lab methods. So the Haney test was developed first and foremost for nutrient recommendations. Soil health came later. Very excited that we're measuring soil health though. We test for biology and chemistry. And the biology tells us whether the chemistry is functioning in our soil and whether the physical aspects of our soil are functioning properly. So that's what we measure. The soil is an incredibly dynamic living system. I said this in the last talk I gave that she sent out, so I don't really need to go over that. But the majority of nutrient cycling is due to the drying and rewetting effect of the soil, and that's what we measure. We want to see why plants are turning greener, what's happening with the nutrient release in your soil. And previous laboratory analysis didn't account for that. So I'm going to go into respiration. Soil respiration, and I'm going to switch between my presentation and some results here, so you're going to have to give me a second. But when we look at soil respiration, we want to have a really good respiration, but it's all based on context. So if soil respiration is first and foremost, microbes take in oxygen and give off CO2, just like we do. So the more carbon dioxide that is emitted when we test for it is more microbial activity, which means that you have a healthier functioning system. This is a breakdown of kind of how we look at respiration, but we need to keep in mind that this is context driven. So if you're in the arid West, you can't compare yourself to somebody in North Carolina that gets adequate moisture, who has warm temperatures all year long. So everything's context dependent, and that's why there's no average on this chart. When there was a big drought the past few years in Kansas, Nebraska, and Colorado, pretty much everywhere. But in the arid West, if you got a respiration of 30, we were pretty excited about that, because that means your soil is functioning. If you're in Iowa, and even though they suffered a drought recently, If you're in Iowa, getting a 30 respiration is probably not a good thing. We want to keep all of that in context. So we're going to start with this nitrogen section. And this is a really good way to explain when you get results, what all the parts mean, and how you can interpret it. So nitrogen. We measure nitrogen using the water extract. Why do we use water? It rains water. If you look in the top left corner of your screen, you will see nitrogen. There's H3A extract and H2O extract. H2O is water. H3A is a combination of salicylic, malic, and citric acid. And we use that because it mimics plant root exudate. And that's the extractant that we use to measure ammonium and nitrate, phosphorus, and potassium, and all of your micronutrients. So in the top left, you have nitrate, nitrogen, H3A extract. That's what most laboratories will test for. If you're west of the Mississippi River, if you're east, for some reason, nitrate, nitrogen doesn't exist there, so they don't measure it, which is a joke. It does. But when you get results from your laboratory, you might get nitrate, nitrogen. What the Haney test measures for is nitrate, nitrogen, ammonium, and inorganic nitrogen. That's the total of ammonium and nitrate, nitrogen. We also use the water extract to measure for organic nitrogen. Organic nitrogen is very important because plants prefer it. It's a part of nitrogen that we couldn't measure before because we didn't have the instrumentation to do that, and now we do. So we're trying to use the most up-to-date methodology that we possibly have and instrumentation we have to see what's actually going on in your soil. Organic nitrogen, as I said, was preferred by plants. They can take it up. They prefer to take it up over inorganic nitrogen because it's more efficient in their processes and building plant tissue and all the things that plants do. Also, organic nitrogen is not readily leachable. It doesn't volatilize like ammonium does. So we want organic nitrogen ratios to be higher than inorganic nitrogen ratios. And as your soil becomes healthier, your organic nitrogen to inorganic nitrogen ratios become higher. So on these results, we see that we have 18 part per million nitrate, about 5 part per million ammonium. We have organic nitrogen levels around 15. So the next thing we look at is the nitrogen comparison results. And that is right next to soil health in that third section down. When you look at your results and you see traditional nitrogen available, that's 33.1 pounds per acre. So that is your nitrate nitrogen in part per million times 2, basically. And that's what you would normally get traditionally from a laboratory. The Haney nitrogen section says that we have 69.2 pounds per acre. So that includes ammonium nitrogen, nitrate nitrogen, and organic nitrogen. The difference in nitrogen available, difference in pounds per acre, is 36.1. So that means that the Haney test found an additional 36.1 pounds of nitrogen available. So what you can do on your soil test, if you do this, let's say you send it to your state lab and you get a difference of 36 pounds per acre when you run the Haney test. You can subtract that 36 pounds from your fertility recommendations because you're not seeing that in your traditional testing. At the time this was calculated, I don't remember exactly what fertilizer prices were 2022, but the nitrogen pounds in dollars per acre that we found was $23. But let's just say nitrogen's $1 a pound. You'd save $36 per acre. And a lot of people are like, do I trust this? Do I feel okay with this number? We are conservative in these measurements. And this is probably something I hadn't said before. If you were to continually measure the soil month after month, you're getting a release of nitrogen. If you have water and moisture, you're getting a release of nitrogen over and over and over again, if your soil is biologically active. So we're doing a conservative estimate on what's going to be available. So people can sleep at night, look at this and say, okay, I'm going to cut back by 36 pounds an acre in my nitrogen fertility, and I'm going to feel comfortable with that because it's a conservative estimate, it's available, and the plants prefer to take up organic nitrogen. Another very important component of the nitrogen part is organic nitrogen release. A lot of people don't understand what that is, but what it means is if we see organic nitrogen in your soil and your soil is biologically active, you're going to get the full entire credit for the organic nitrogen that's available in your soil. So this soil right here is very, very active, and I'll go into that later, but we give this person an entire release of nitrogen in the soil. They get full credit for all of the organic nitrogen we've seen. If you have a soil that is less active and your biology is stunted, we're going to dial that back and say, okay, not all of your nitrogen is being cycled, so we're not going to give you full credit for that. So when we're looking at your soil and your results and your conditions, we can say, okay, you have this much organic nitrogen available. You have a lot of biological activity. We know this stuff's going to be chewed up by the biology, and it's going to be released to your plants. If your soil's hampered because, let's say, flooding, or you don't have enough airspace, water space, which would be flooding, no biological activity, we're not going to give you credit for that. I think that a lot, and I've been asked this a lot lately, like, why would producers feel comfortable using these results? Just know that we're accounting for what's there, what's actually happening in your soil, and we're giving you the best credit we can possible without damaging yield. The one thing we don't want to do is give you a yield hit. And to date, we have only had one person say, I followed your recommendations and I didn't make my yield goal. And when we asked them why, it's because they didn't get any rain the whole entire season. So regardless of what they put out, they're not going to meet their yield goal. We can't control the weather. So I'm going to move on to phosphorus now. And since, how many people are on here, Yazzie?
Like 65.
65. Okay. That's more than before. I was going to let people ask questions, but not yet. Okay, phosphorus. With the Haney test, we use the H3A extract to measure phosphorus. And the reason this is so important for phosphorus is because standard tests use Bray, Olsen, Malick. Malick is touted as a universal extractant. Malick extract, which is what most of your laboratories use, is a pH of 2.5. It hits the soil in the laboratory. We hit it with Malick at pH 2.5. It changes the pH of the soil down to 2.5 or close to that. And we start releasing calcium phosphates, iron and aluminum phosphates, which aren't actually plant available at the time. What H3A does is that it's not buffered. It's not a buffered extract. It's trying to mimic what the plant roots actually pump out into the soil when your plants are growing. And it mimics the soil pH. So we're not altering the conditions, and we can see what's actually available in the soil to your plants. So a lot of people have said or asked if the Haney test always recommends lower amounts of fertility. No. In Texas, we have pH 8.385 soils. We have massive amounts of calcium. The whole entire state is like phosphorus deficient because they were using, or they are still using, the H3A extract to determine phosphorus fertility. And that is underestimating the amount of phosphorus that our plants need here to grow in Texas. If you have a very low pH, your recommendations from a laboratory might say you don't need as much phosphorus because all your phosphorus is tied up in iron and aluminum. So we want to actually measure what's going to happen in the soil. H3A is a mimic of plant root exudate. They're going to dump it out, solubilize nutrients, your plant's going to take them up, and that's what we're trying to measure. So we have total phosphorus in parts per million on this example with 16.1. We also measure organic phosphorus. The difference between inorganic and organic phosphorus in the way that it's measured, it hasn't been measured in the laboratory before. We take, I don't know how in detail you want me to go, but we measure inorganic phosphorus using a colorimetric method. And then with organic phosphorus, we're like blowing up the whole sample. So we can see the total amount of phosphorus that is available. And by subtraction, we can get organic phosphorus. Again, organic phosphorus is going to be taken up by plants. There's not just nitrogen, phosphorus, carbon, anything by itself. Plants are taking all of these compounds up in whole. And so anytime that the microbes are chewing on carbon, they're chewing on nitrogen, phosphorus, iron, manganese, aluminum, all of the different, magnesium, sulfur, anything you want. And so we want to measure all of those things are going to be available to the plant. And that's where your organic phosphorus number comes in. Similar to nitrogen, we will not give you a credit for organic phosphorus unless you have microbial activity. So the soil does insanely amount of microbial activity, but they get all of the credit from the organic phosphorus that we measure. The other soil measures section, it's very similar to what standard testing does. You get your soil pH, a buffer pH, soluble salts, excess lime, and then soil organic matter. Okay, so when you look at your results from a laboratory, you want to ask which lab method that your laboratory uses, and your results are going to vary based on that. We have attempted and tested all the different methods. The way that the Haney test was developed is that we tested different extracts and we compared plant available phosphorus using a very tried and true method called iron and aluminum oxide strips to determine it's the gold standard for plant available phosphorus. It's not realistic to use in a laboratory setting. It takes too long. It's not cost effective, time consuming, all those things. So we compared the Haney test using the H3A extract to iron and aluminum oxide strips and found that malic 3 extracts around 2 times what is actually plant available. And the Haney test, H3A extract, measures pretty much a 1-to-1 ratio of what is actually plant available. So these are Russell fields. He put in a trial last year. He followed the recommendations using the Haney test on the green stripes. He followed the recommendations using Malic 3 in the yellow stripes. And then the red section was the Haney test plus his Foliar program. And I'm not going to go into that part. However, the Haney test, where he applied nutrients according to the Haney test, showed tremendous growth early on. Soybean plant on the left is using Haney. Soybean plant on the right is using Malic 3 recommendations. And the big difference here is phosphorus and potassium mostly. And I know I don't have sound on this, but when you look across the field, you can see a bump in growth. So he's standing in a Malic 3 plot, and where he just pointed is the Haney test plot. So plant growth is definitely higher, different, much larger. And that's what we're going for. We want to maximize your production by giving you the optimal fertility recommendations. Yeah, have them faster. Nice. We went out there and Rick popped up in the field because it was a Haney plot. Not funny. I don't like it when people can't laugh. Anyway, the plant on the left in his left hand is from a Haney plot, and the plant on his right hand is from a Malic plot. We could see even at this point, at the end of the year, there was a big difference.
The Malic 3 was 38 bushels. The Haney was 54. So essentially the Malic 3 was like 23 pounds of nitrogen, 60 pounds of phosphorus, 60 pounds of potassium, and 9 pounds of sulfur. We reduced potassium by 30 pounds. We reduced sulfur by 2 pounds, but we had to increase what she was talking about, phosphorus availability. We had to apply 10 more pounds of actual phosphorus. So we reduced total fertilizer applied by 57 pounds. So we applied less product, but we did have to apply 10 more pounds of phosphorus, which I think, you know, in the grand scheme of things, our net return was right at $180 more profit with the Haney test than with the Malic 3. So we started doing foliars 5-6 years ago. You know, one of the things that we were told by our university is foliars didn't pay, they didn't work. If you're going to go out there in the middle of summer when it's 100 degrees and foliar apply, you know, noon, you're probably not going to see much benefit. I mean, one of the things I will say is we typically foliar 8-9 o'clock at night till 4 in the morning, or either a really early morning before we get over 85 degrees. And, you know, working with Meredith, talking about the algae, that's one of the things we've seen with the in-soil algae is the effectiveness of our foliar passes. So you can see here that we hit beans at V4 with a pint of soy sauce, some sugar, a little bit of calcium, the in-soil, some molybdenum, and then we came back at R3, and it was pretty much close to the same, just a higher dose at that point because we had a larger plant. But with our foliar program, we were able to take the Haney test from 52 bushels to 86 bushels per acre. So, I mean, if you are lacking in some, you know, nutrients in the plant, and you can make, you know, a $10-15 foliar spray, even $20, your money ahead. At least for us, we have been, you know, our farm average the last couple of years has been in that 80 to 85 bushel range for beans.
So what's so cool about his field is that right now we have a soil health score. I'm going to talk about the soil health section. Soil respiration is a measure of microbial activity. as I said before, microbes take in oxygen and give off CO2. So the higher your respiration, the more active the soil is, and the more nutrient cycling you have. So these are recent results from 22. They're probably better now. His soil respiration is 414 part per million. And so nobody needs to strive for that necessarily, because that's really insane numbers. But when Russell started, we dug up the results yesterday of this field from 2013. These tests were done when Rick still worked at USDA, ARS. He had developed it when he worked for ARS. Russell came down and hung out with us, and we did tests for him. And his respiration on the same field that we're talking about right now that has 414 part per million, 415 part per million respiration, when he first started, it was 55, which is still a great number. But over the past, 10, 11 years, because of the practices that Russell has been doing, which is no-till, he heavily manages his fertility. By heavily manage, I mean pays attention to every single detail. He uses biological products. He uses cover crops. He does all of the wonderful things. His respiration has increased from 55 to 415. So what does that do for you? That gets you the champion of corn contest. So he broke the corn yield. So when people talk about, is this soil biology working? Does this testing matter? Yes, it matters, because we can see what we're doing with our management practices year over year over year. And over time, you can really tell that you're amping up your soil biology and your nutrient cycling, even just based on this one number right here, which is 1 day CO2. I'm trying to zoom in here. So we're going to look at his, and these are like old results before we had PDFs, like old stuff. His organic nitrogen numbers were 43.1. His inorganic nitrogen was 35.2. He had really good numbers to start with, and not a lot of people do. But over time, as we improve practices, and this is one of the important things about doing the Haney test, is that you can monitor whether things are working or not, whether your management practices are improving what you're doing on your soil, and whether they're working for you. So we're looking at the soil health section. His respiration is 414 part per million, which is tremendously improved from when he started doing regenerative practices or farming, period. The organic carbon part per million. So this part is very important. A lot of people only look at soil organic matter, the total soil organic matter, which we have here under other soil measures. The water extractable organic carbon part is the part that microbes see. It's the part that microbes chew on. It's what they eat to release nutrients. And so we want to see this value increasing over and over and over again. I've seen soils with, just today, 1% organic matter, and their organic carbon, the water extractable organic carbon part, was so high, 300 part per million. It was insane. And what's happening in the field I saw today is that wheat was growing. So wheat's growing, it's pumping out plant root exudates, it's pumping out that organic carbon and feeding those microbes, and they chew on that, and they give nitrogen and phosphorus and magnesium and sulfur and all that back. So we want that to increase. The MAC percent is something a lot of people don't maybe understand or haven't looked at. MAC is the microbially active carbon. So you can have a whole lot of food source in the soil, But if which is water extractable organic carbon, but if your soil is compacted or flooded, or you don't have good aggregation, or the temperatures are off, your microbially active carbon is much lower. So at values below 100%, let's say 50%, we're worried that the microbes might not like the carbon source in your soil. You might have compaction issues, you might have infiltration issues. They're not getting all of the different parts that they need to live and survive and cycle nutrients. And what's cool to me is that when you start incorporating cover crops and diversity in your system, that your MAC percent goes up, your water extractable organic carbon usually goes up, and it's because your soil is more resilient. So the more cover crops you have, the more diverse they are, they're putting out different extracts and different carbon sources to feed different microbial communities. So your soil is very resilient. And we want that MAC percent to increase and increase and increase. However, in Russell's situation, his MAC is 230%, which is insane. So when you get over 100, 125% MAC, what we see with our farmers is that their soil is so active, they have a hard time keeping residue on the surface. And that's not really a problem. You just have to manage for it. Or I don't know, Russell might think it's a problem, but you have to manage for it. So there's a balance in everything, and that balance is in the carbon to nitrogen ratio. And so right here, the carbon to nitrogen ratio is around 12. We'd like it to be between 12 and probably 16, 17. You want to have a balance in that field. And so I know I have to wrap up soon, but this part is where this soil health section is where we really can look at your soil. We can see if you have good air and water infiltration. You can see if you have an adequate food source for the microbes in the soil. We can start to look at problems. Is your soil imbalanced? Is it imbalanced? And start making some cover crop suggestions. One thing before I stop is that the cover crop suggestion is very general. When you get one from a Haney test, it'll say percent legume, percent grass. The legume part is legumes and brassicas. The grass part is high carbon to nitrogen ratio plants. So if you have any questions about that, just feel free to reach out to us.
Thanks, Liz. We have,
okay.
yeah, we have a couple of questions coming in, so maybe we'll start with those and go from there. So we have someone asking, and I know that Regen Ag Labs does this, but how do you find agronomists that have expertise in reading the Haney test and giving sound recommendations?
There are some out there, but if you want to know, like in your area or someone near you that specifically you can trust and rely on, just reach out to us and we'll help you find someone. Or we'll do it for you.
Will a soil test taken late fall versus late spring in the same location have differing readings?
Yes, 100%. Because biology is constantly moving, it's constantly changing. If you take a sample today and then take one 3 days from now, it's going to be different. And thank goodness. And that is one of the complaints we've gotten. Like, this stuff isn't static. Like, we're not getting static enough readings. No, you're not. It's because biology is constantly changing. It's constantly moving. And we want to see what's happening in your soil right now, or at a time where you can make management decisions. So hopefully, yes, it will change.
Great. So feel free to unmute yourselves, y'all, or put some questions in the chat for Liz, and we will go through those for the next 15 minutes or so.
You want me to add to that, Liz?
Please.
So if a producer's looking at a fall versus a spring sample, number one, you have to take into consideration what crop they're harvesting. So just take corn. If that producer, they're going to have to use a little bit more of their mental capacity, because you're going to have 100 to 200 pounds of potassium tied up in corn stover residue that's not going to be broken down yet. There's going to be a lot of nitrogen, phosphorus. You're going to see, if you pull one in the spring versus one in the fall, you're going to see a huge difference in plant available nutrients on the Haney test for one. So a fall test, say 2 days after harvest, is probably going to call for a higher fertility rate than a spring sample. But we have a very good understanding of what units or what, nutrients we have in that residue. You can subtract that off. We've done that before, and that's 95% accurate. And then also, one of the things you're going to miss is if you do plant a cover crop and you're building organic nitrogen, or you're seeing residue breakdown, you're going to miss a component of the nitrogen on the Haney test that you'll pick up in the springtime, right at planting or right after planting, that you'll not see that in the fall sample typically.
Thanks, Russell. And there's another question to maybe add to this for either of you. Is one Haney test per year adequate? And along with that, what would be the best time to take a single sample in a year?
That depends on your goals. If you're looking for fertility recommendations, pre-plant, do it then. If you want side dress recommendations, do it then. If you're monitoring your soil health year over year, and you just want to see if you're improving or not, do it at the same time every year, meaning same temperature and moisture conditions.
And I think the only thing to add to that is, you look at like Wisconsin, Minnesota, some North Dakota, South Dakota, their soil temperatures are barely going to be peaking 50 degrees at planting time, because a lot of them will start putting in, seed when it's still a little bit colder than it should be. And we do have a lot of producers that they'll put out, a little bit of their NP and K and some of their micros at planting time. They'll wait, 2 weeks in, 3 weeks into the season when there's, temperatures are 55 degrees, 60 degree soil temps. And then they'll make another nutrient pass based on, what they see there. So you may have to make some different adjustments for northern states, but yeah, absolutely. I mean, even if a producer's just looking at, how do we do better nitrogen management, like the test, Liz showed of mine, we don't get nitrate results like Liz said here in the East. So, we're spending $1.25, $1.50 an acre to run these Haney tests. And, based on our traditional testing, we're picking up 60 to 80 units of nitrogen that we're able to cut back. And like this year, it's running, 60 cents a unit. So, I mean, you're still talking about $40 an acre in savings just looking at nitrogen, even if you don't take a look at the, the P and the K. But for us, phosphorus management, the Haney test sometimes does call for a little bit higher phosphorus than Mealic 3. And just adding that additional 5-10 pounds, like you saw in that soybean study, you're talking about spending less than $10 an acre in additional phosphorus that plant truly does need. And it's making, $180, $200 more an acre in yield. That's where we're really seeing, it outshine what we're seeing with our Mealic.
Great, thanks, Russell. Along with the sort of logistical side of things, someone's wondering how many samples are recommended per 80 acres or per a different size field, and how does the composite testing work? So how do you composite them, and what's the looking at them individually or per soil type look like?
Okay, so if you're doing 80 acres and you're not prescription applying, take one composite sample for that 80 acres. You can do it on smaller acreage and that's fine. But you would want to take at a minimum 10 cores from 0 to 6 inches and combine them together. And the way you do that by soil type is, let's say you have, for a very simple example, you have 100 acres, 80% of that is one soil type, 20% is another soil type. Take 8 subsamples from that dominant soil type and 2 from the subdominant soil type. So you want to parse it out based on your soil types, and you can access those soil types from USDA, or you can use EC mapping, you can use yield mapping, you can pick whatever, but get a very representative sample of the whole entire field so that you're getting a good representation of what's actually going on in that whole field.
Great. Are there any other indicators that could enhance the impact of the Haney test, such as soil organic carbon?
So when soil organic carbon is great, and we love that stuff, and it helps with aggregation and water holding capacity, but it's not the biologically active part of the soil. I've seen soils with 1% organic matter have very high microbial activity, and that's because the microbes like what's in the soil. There's a plant growing, they're cycling nutrients actively, and that's what's most important. So we always want to build organic matter for sure, and that's what you do through your practices. But what's most important is what's available to the microbes to cycle, and that's that water extractable organic portion. Additional tests that you can do to see what's going on in your soil. You can do a phospholipid fatty acid test. You can see what your bacterial to fungal ratio is. You can start to learn more and more about how all of that's working together. You can do metagenomic testing. You can do plant tissue testing. The more testing that you do, the more knowledge you're going to have. I asked everybody, a lot of regenerative farmers that I know, including Russell, a couple of years ago, like, what's the big difference between being an a regenerative farmer versus conventional farmer? And so conventional farming, there's a lot of, I'm planting at this date and I'm going to put my butt in the tractor for this amount of time. Regenerative farming is thinking about and testing what you have, what you know you have, and then making a plan around that. And so it's just a little more foresight and thought behind the process.
Great, thanks, Liz. There are a couple of questions coming through for Russell, if you're cool to answer some of these. Mike's wondering, which cover crops do you now use since you have such active soils? And are you willing to explain some of the intent for each of the ingredients in your biological foliar plan?
So I guess the first part on cover crops, we treat the cover crops as a nutrient path as well. And by what I mean is, just take small grains. We use 3 different small grains every year in our base mix. It's cereal rye, triticale, and oats. Like Liz said, having residue coverage is an issue for us later in the season. So that's why we use cereal rye. It'll be there at harvest. Carbon and nitrogen on it's probably going to be 80 to 1. Triticale, it's probably going to be 50 to 1, and oats are around 35 to 1. And so those oats are typically breaking down, V3, V4 corn, that's releasing nutrients. The triticale is breaking down, anywhere from V8 to VT, and that's another nutrient release. So we're essentially side dressing nutrients to our crops through the cover crop residue. We also try to balance the carbon and nitrogen ratio. I think that's where a lot of farmers miss the point. They'll talk about, planting into green rye and they had the allelopathic effect. And I always ask farmers, if the corn germinated, it wasn't a allelopathic effect. You just soaked up all the nitrogen because you had too high of a carbon to nitrogen ratio. So we shoot for one, essentially like our area is getting 40 inches of rainfall. So we're shooting for a 40-to-1 carbon to nitrogen ratio on the cover crop residue. And then we're balancing that out with, clovers, vetch, winter peas, brassicas. And that's kind of how we do that in the field. What was the other part of that question?
Are you willing to talk about some of the intent behind your foliar plan, the ingredients in it?
Yeah. So when we started seeing higher yields, even though we have biologically active soils, sometimes we see potassium issues. And so the soy sauce is a foliar product made by Wind Biologics, and it's a 5012. It has potassium acetate in it, which is the most readily available form for us to foliar feed. So we use a lot of potassium acetate. A quart to the acre of that cost us about $4. So it's not relatively expensive. It's got fulvics, it's got humics. We're putting sugars in. That was the Sweet Success or the Wind Sweet Soil. Either way, we're using sugar to bring up the Brix level to fight pest. When we started working with Meredith, one of the things that kind of intrigued me about the in-soil algae is it had a micron size of 5 microns, which is extremely small. And it's actually acting as a carrier to help pull some of those foliar nutrients into the plant. That's why we put it in our foliar pass this year. We're doing some studies for Meredith and Tucker, where if we do have to do some fungicides, or some neighboring farmers in the state, if they do any fungicide passes, we're going to treat it with algae as well to see if we see a higher efficiency of a fungicide pass where we can actually cut the rate back. Because algae is a lot cheaper than fungicide. And then, we're just looking at what the plant needs. I mean, we're taking tissue samples, and then whatever we see as a deficiency, that's why we added a little bit of molybdenum. Typically in our low CEC soils, we're always deficient in boron. So boron has always paid for us. A quart of boron's about $3, $4 an acre. And that's really helping us, treat those deficiencies and maximize that plant.
Thanks, Russell.
Speaking of boron, I have a question from Jim. He asked, how many square feet of growing cover crop to send in a sample, and does the test look at boron levels?
On the cover crop analysis?
Yeah. How many square feet?
So you can do it
also talk about soil boron.
Okay, so you can do, so talking about the cover crop nutrient analysis, you can do 1 square foot and do it 8 times across the field randomly, so you get a good representation. We just picked up an NRCS standard back in 2013, and that's what we've always ran with. And we do 2 square feet, and we pull 4 of those across the field. So 8 square feet is what we're going for in total. And when we send that in, we're getting, NPK, all your micros, your macros. If your carbon and nitrogen ratio on the cover crop, like for us, is 40-to-1 or less, we're taking 70% of the nitrogen, and then or 75% of the nitrogen, and then 70% of the P, the K, and all the micros that are there. So we can also take that as a credit. When we're looking at soil boron, for years we were pulling tissue samples and we were applying what the Melic 3 said to do. And, Melic 3 was saying, put on 1/10 of a pound, 1/4 of a pound, and our tissue samples were always below adequate or deficient. And so Lance told me about what they were doing. They're doing hot water extraction. I think we're paying $4 to have them run that hot water extraction. And it's coming back 2, if not 3 times higher than what Melic 3 does. So, when Liz was talking about Melic 3 overestimating phosphorus and what we saw with that soybean plot she sowed, it's even doing the same thing with other nutrients. Boron's just one of them we've paid attention to. So like we do the hot water extraction, and instead of applying 1/10 of a pound or 1/4 of a pound, we're applying a half of a pound, sometimes 3/4 of a pound. And like I said, we're spending an extra, $3 for every quarter pound. So even if we're spending, $3 to $6 an acre for that additional boron, it's what the plant actually needs, instead of what a false, reading is giving us on the mealy test.
Thanks. We got some more in the chat. Terry wants to know, are the results from different labs consistently accurate? We are sending ours to AEL in Australia. Does the delivery time from sampling have an effect on the sample? And also thank you for your explanations.
Yeah. So laboratories, they're going to vary wildly. And there's only one laboratory that does it the way that it's supposed to be done, and that's Regen Ag Lab in Nebraska. They're the ones that have kept up with the technology and not branched out and modified it themselves. Delivery time from sampling, it depends on temperature. So if you're going to ship samples to, let's say you're shipping samples from Australia, you're going to might do like a 2 or 3 day shipping, especially if it's hot outside. Heat, extreme cold, that's going to affect samples. It doesn't mean you have to put them on ice or refrigerate them or anything when you send them in, but don't leave the samples on your dashboard. Of your truck. Don't throw them in a barn and forget about them for several months. Try and do it in a timely manner. And as far as people trying to help understand the soil test results in Australia, just call us and we'll help you.
Nice. Okay, David. Oh, by the way, everyone, it's 8:03. So if you are able to stay on, we definitely have some more good questions in here. But if not, thanks for coming, and you'll get the recording tomorrow. Okay, David wants to know, is there biomortality in time taking sample to lab, receiving sample and shipping? Oh, is there biomortality in the time taking the sample to the lab and receiving the sample? And
yeah. So probably, but so the way that microbiology or the microbes work in the soil is that the soil is dried and re-wetted, and that happens in the field and that happens in the laboratory. So what happens is you dry the soil out, there's a whole bunch of microbiology that dies, and then once you re-wet it, the living microbes cannibalize on their brethren and reinvigorate and start living again. And that's what we measure. And so it's okay that some of it dies because it's still a measure of your microbiology. You're measuring the microbial biomass in the soil, whether it's living or dead, because everything moves and transitions as it goes. Again, though, please try to not, put it on a dashboard and overheat it or anything like that. Try and keep it as good as you can.
Yeah, like you'd want to be.
Yeah.
It makes me laugh to always think that there's a microbial war going on in the soil.
Yeah.
Yeah.
It's crazy under there.
That sweet smell when you till genocide.
Okay, Sean wants to know, what test indicators are you seeing that are making a difference in soil health when using a biological product like Soil Algae?
I mean,
what I've seen. Oh, go ahead.
No, go ahead. That's all right.
Go ahead.
I was just going to say, I mean, we're paying attention not only to the Haney test, looking at the CO2 respiration, but we're also paying attention, the PLFA, like Liz mentioned, what are we seeing with total microbial biomass, fungal to bacteria ratios, We even started working with a company called Rise Bio that's doing metagenomics. And so, we've got samples from last year, with biologicals and without, with all three of those tests. And, it's in the early years, you're going to do a lot of testing to figure it out. And then as you progressively go down this path, you still do testing, to verify what you're seeing, but you can rely more on a shovel and what your eyes are seeing more than, pulling so many tests back to back.
I mean, at some point, if you observe what's happening with your testing and over time, depending on how intensive you want to be, well, like Russell always tells people, if we're still having to consult you 5 years out, then we haven't taught you anything. And it's the same thing with soil testing. There's going to be a point where you know what's going on and you understand it and you're in connection with it, and you're not going to have to worry about it. As far as test indicators go as well, the PLFA results that we've seen from using in-soil algae have been very, very interesting to me. It brings back protozoa, which are key to soil function. I've seen most soils have 0 protozoa, but when you add algae, it's there. But soil testing is not the end-all be-all. And so we did a study with Dr. James White, and we saw what the algae was doing within the plant, which we would not have been able to see otherwise. So all of it goes together hand in hand. It's a very complex system. So we're trying to look at all of the different results together: tissue, lysophagy, soil respiration, everything.
Cool. Thanks, Liz. It's 8:08. We're gonna hop off the call. If anyone has more questions for us, for Liz, for Russell, we're happy to make connections. So please reach out to us. We appreciate you coming. And the last thing I'll say is we have a special deal that we're doing with Regen Ag Labs for purchasers of in-soil algae getting a free Haney test, and some people qualifying for an interpretation of that Haney test. So that information will be in the follow-up email tomorrow with the recording. So look out for that. And we'll be in touch. Have a great night, y'all.